Good morning. Let's have a class. And this will be our last class, and before the exam one. And next week, Tuesday, we will talk about the detail of the review questions. So on the eCampus, please go ahead to do all the practice questions. And I will talk about every single question on next week, Tuesday. And then, the key will be posted on the eCampus on Tuesday evening. And Wednesday, I think my student, Carly Long, will do some of the review section for you guys if you need it. And Thursday, next week, Thursday, 11 o'clock here, we'll do the first lecture exam. Okay, so please do these, all the review questions. You can have a group study in the library or have some people gather together to find out those questions. And you can see those questions basically is based on the slides, so you can easily find some. Okay, and if you need to go back to watch the videos, and all the videos are here, and this is I'm missing like a lecture 6.1, so I need to add it. So I need to go back to add this lecture 6.1. So I'm just uploading right now. Okay, let's finish talk about right here. So we finished started talking about the factors affect bacterial growth, and we talk about the temperature, and we talk about pH. We talk about water activity. And then the last one we left didn't talk is requirements for the oxygen. Now when you look at the slides, it looks like complicated. The whole slide is filled of all the pictures and the letters is confusing you. So let's make it clear and start talk from easy to little bit difficult stuff. Okay, so that's called the requirements of oxygen on bacterial growth. So first of all, we need to know, based on the requirements of the oxygen for bacteria, we can simply categorize them into three groups. Number one is aerobic bacteria. I will write number 3 right here. It is anaerobic bacteria. Now, there's one thing in the middle is facultative. So those are the 3 categories you should know at the beginning. So one is aerobic bacteria. Aerobic bacteria, which means a bacteria must use oxygen to survive. Without oxygen, it will be dead. A good example will be Pseudomonas. Pseudomonas aeruginosa, Pseudomonas fluorescens, it's a sporage bacteria. Then we want to talk about at the bottom. Anaerobic bacteria, which means bacteria cannot survive in the presence of oxygen. In other words, exposure to oxygen will kill them, will kill bacteria. The first one must use oxygen, the bottom one cannot use oxygen. The oxygen going to kill them. But then how about the middle? Okay, sorry, by the way, anaerobic bacteria, Clostridium. Clostridium species will be a good example. How about the facultative bacteria? bacteria can grow with or without of oxygen, but grow better with oxygen. Now, what are the examples? Most of the bacteria, I would say majority, pathogens. Let's say Salmonella, let's say E. coli, 0157H7, and the Listeria monocytogenes. All these major foodborne pathogens, they are facultative bacteria. Okay, so this is the three major categories: aerobic bacteria, facultative bacteria, and anaerobic bacteria. Now, based on these three major categories, we have some of the things really in the middle. Therefore, it comes out with, somewhere here, it comes out with so-called microaerophilic bacteria. What this means? This bacteria need oxygen to grow. However, the oxygen amount cannot be too high. It's tricky. So we also call these like a fastidious bacteria. Very picky. Some of them are very picky, you can understand. And they require usually 2 to 10% of oxygen. However, if the oxygen more than 20%, will kill them. It's very picky. That's called a microaerophilic. Which bacteria is that? We mentioned Campylobacter. Let's say Campylobacter jejuni. It's a major pathogen on poultry meats. That's a microaerophilic. So I will write 4 right here. How about the last one? Aerotorrent. That's called aerotorrent. Aerotorrent bacteria. This means the bacteria grow equally well with or without oxygen. It doesn't matter. You're going to do the same thing. That's the last school. I can't name any of the regular aerotorrent bacteria to you. Some of them are environmental microorganisms. And I looked at the list, and none of them I know, I've heard of. At least, so I just tell you there's an example. Now we talk about all these categories. Then we're going to move on to why some of the bacteria needs oxygen to grow. Why some of the bacteria cannot use oxygen to grow. But this is related to something we talk about is electron transport chain for the bacteria to transfer oxygen and then finally use oxygen as a final electron acceptor. So this is a little bit biochemistry. You have electron transport chain, ETC. What it looks like? NADH goes to FAD. Then is a plus, is FMN and FES. So we go to COQ, coenzyme Q, then go to cytochroma C, cytochroma A. Sometimes we go cytochroma A3, and the final is oxygen. Okay, this is the key. So the electron going to be transferred and delivered from the electron carrier in this electron transport chain. So first of all, if it's bacteria, they are using oxygen as a final electron acceptor. So they deliver to them. And you know that there is this one accompanied with repel of these protons outside of the cell membrane system, then generate so-called proton motive force, and then they will generate ATP finally. Okay, we'll talk about that later. That's one thing important. But at the same time, you need to know, you have the oxygen, and oxygen has a very special structure of the electron in their outer atom. Just 2 or 3 available. Oxygen sometimes like this. Here is a big deal. It's not stable. So what happened? The oxygen could be working with an electron, then become what? A superoxidase, something like that. We call it a superoxide. Or superoxygen. We just say it simple. Superoxygen. And then this superoxygen, together with a proton, we mentioned about this proton motive force. And then with another electron, they will generate another chemical. What is that? H2O2, hydrogen peroxide. And unfortunately, this guy, superoxygen and hydrogen peroxide, those are toxication for bacteria. So in other words, said very simple, during this electron transport chain, they needed to generate energy, ATP, but at the same time, they're going to generate this toxication material, superoxygen or hydrogen peroxide. So what are we going to do? Once we have this guy here, what are we going to do? We have to hydrolyze it. So how to hydrolyze? There's some other proton there. So we use it, become then we release oxygen. This needs an enzyme. The enzyme called SOD. What's the full name of that? So superoxidase das mutase, or superoxide das mutase, SOD. Then you have this hydrogen peroxide. The hydrogen peroxide is not good. So what we have to do, we need a catalyst to hydrolyze it, become oxygen and water, and this will be released. Then it will be safe. So that means if bacteria need oxygen, they must have these 2 enzymes, SOD and catalyst. Therefore, why some of the bacteria must use oxygen? Because they have SOD and the catalyst. And in other words, why some of the bacteria cannot survive in the presence of oxygen? Because there is no SOD, no catalyst. The reason is that if the oxygen is there, they will generate superoxygen and hydrogen peroxide. They're going to kill the bacteria. You don't have those enzymes to hydrolyze them. So that's caused a problem. But how about these facultative? Microaerophilic and aerotolerant, well, somewhere between that. Then you look at your slides. You will see it. Facultative. Yeah, SOD is there. Catalase is there. How about the microallophilic? Well, SOD is there, and the catalase maybe, maybe not. Maybe there, maybe not. How about the alloterant? SOD is there, however, there's no catalase. That's a major reason will cause a difference because of these presence, absence of the two major enzymes will let the bacteria to survive in the oxygen and does not survive in the oxygen. So what's going to happen when you express it in the bacterial broth medium? Look at your picture. All the aerobic bacteria, they grow on the top because they need oxygen. In other words, all the anaerobic bacteria, majority of them is at the bottom. Okay, so we can draw the picture right here. Aerobic bacteria most likely is on the top because it's exposed to the oxygen. Anaerobic, lots of them is at the bottom. That's anaerobic. But how about the facultative? Yeah, something in the middle, something at the bottom. Basically, averagely covered the inside of the tube. That's facultative. Facultative, F-A-C-U-T-A-T-I. How about the microallophilic? Sometimes a little bit bottom of the top. That's microallophilic. And we said that it needs oxygen, but a very limited amount. How about allotorrent? Allotorrent, basically, you're not going to see much difference. It's averagely covers the whole tube. This is allotorrent. Okay, so that's, we see the arrangement of this bacterial density inside of the tube. We also can get a general idea what type of the microorganism it is, based on the oxygen requirements. So the key thing is that 5 different categories. How you learn it? Remember the first 3. Then talk about the microallophilic and the allotorrent. Now, why behind it there is a big story? Because of the enzyme, SOD and the catalase. With or with that, will depends on their requirements of the oxygen. Okay, the next one, what we want to talk about is how we got a cultivation anaerobic bacteria. There is 2 methods we can do. The first method is relatively easy, however, it is expensive, which is anaerobic chamber. Look at the left side, it's the anaerobic chamber. Okay? If you wanted to do this anaerobic chamber, you have to connect it, have a good connection with the commercial company, and to purchase one. And there is 2 tunnels, you can use your hands put in, you can do it inside. And there's a pump outside, it will automatically extract all of the oxygen. But this chamber somewhere, $25,000. It is expensive. And also you need to have oxygen supply sometimes. And it's expensive. Most of the time what we did in the lab is using a jar. It's a pretty nice small jar. Okay, it's a small jar, like 5 liter. Sometimes 2.5 liter. We call it anaerobic jar. So we can talk a little bit about that. The story behind it is very interesting. Inside, we usually put a gas pack. It's a commercial gas pack. Okay? Let's say we put agar there. The agar cannot hold on too much. And sometimes 10 to 12, 12 hours. It's a small jar, not that large. So we put a gas pack. And the gas pack, we're going to fill in with 15 ml water. So what they're going to generate, this will generate hydrogen. And the hydrogen will react with oxygen originally inside of the jar and it becomes water. And we want this reaction to happen really quick. So on the top, underneath the bottom of the lid, sometimes we have those catalysts under the name called palladium. We put a catalyst there, let this reaction happen more quick. Now finally, how do we know the oxygen is already gone? We need some of the oxygen detector or indicator. And most of the time, we will add a strip of methylene group. We add methylene group. Methylene group. If this methylene group becomes colorless, that means the oxygen is already gone. If the methylene group becomes colorless, which means there's no oxygen. Another chemical we can put is resazurine. And when the resazurine has become colorless, from pink to colorless, which means oxygen is gone. Okay? Lots of the time, we also need to put a paper towel inside somewhere because a lot of the water going to generate, it will cause the condensation of those agar media. So we put a paper towel inside to absorb all those extra water. So that's what we did for the anaerobic jar. And it's very interesting. And this jar usually is also expensive, somewhere about $200. Even this small tiny jar, sometimes $200. And you have to buy those gas pack, and that could hold 10 to 12 hours. But compared to these chambers, that is much less expensive. Okay, so that is for the anaerobic bacteria that grow. Okay, next one, we want to briefly mention one special type of the bacteria class. It's called biofilm. We mentioned it briefly, but right here we want to talk a little bit more. And after that, we will be mainly talk about the big topic here is for the bacterial culture media. There is a lot we could talk. Okay. So first thing we want to talk about the biofilm. So what is the biofilm? Biofilm said very simple. I would say this is a matrix of a group of bacteria on, I would say, food contact surfaces. So I'm a food microbiologist, and what we care about is really the food processing equipment and contact surfaces to have the biofilm. So let's say the bench. I was cutting the meat. I was preparing the beef patties. And I see the other bacterial biofilms, a matrix film. That's what I'm interested in. Okay? Well, of course, it could be on the floor, could be in the high tunnel, inside the chamber, could be in the glass, could be in different material. But we don't care about that too much. I really care about the biofilm is on the food contact surfaces. So this is what's going to happen. We're going to briefly introduce to you. And the picture in the slides is a little bit messy and complicated. So any of the food contact surfaces, more or less they have some water. So here is the key thing. environmental. As long as there is water, the bacteria could grow. So the moisture really have to control the bacteria growth. So the same thing for fungi and yeast modes. Whenever you see some area of very humidity or have a water dropping, very possible you have a bacteria there, as long as there's some of the water. Second thing is that any of the food contact surfaces, more or less, you have some limited nutrients. Those limited nutrients can let bacteria grow there. Although it is not as many as we see in our bacterial culturing media. But we're going to have some. So what's going to happen? Then the bacteria are going to go there. Okay, a couple go there. Now once they go there, they're going to connect it with each other at the beginning. How they connected it? This is a physics person, some biology physics person do a research. They find they are connected with band, vault, force. You heard about that. Is there ionic bonds, hydrogen bonds, is there covalent bonds, and they could be band, vault, force. It's a loosely attached. Okay? Now, of course, some will be released. They're not going to be connecting the line, so we want to dislodge. So they release it. They got it. Okay, left some of it. They're going to connect it more and more. More and more. Something like this. Connected. Why they can do that? Because there is a very important stuff there. We call it a cell-to-cell communication. And we have a terminology for that. We are called chlorine sensing. And they are connected, also rely on a chemical. This chemical kind of called lactone, but it's not really a basic lactone. It is N-acetyl hormone serine lactone. And we have an acronym called AHL. They rely on this chemical to do the cell-to-cell communication. And this chemical first time is isolated from fibril. Fibril is a pathogen in the seafoods. And in the seafood